Hi, I'm Song Lee. I'm an associate professor in Department of Bioengineering at UC Berkeley.
And I'm Kyle Kurpinski. I'm a PhD candidate also in bioengineering at UC Berkeley. And today we're going to present a protocol for mechanical stimulation of stem cells using cyclic uniaxial strain.
The research in our lab focuses on cell and tissue engineering, especially in the cardiovascular system. And we're interested in the mechanobiology of stem cells because there are a few potential applications. One is that we want to understand the effect of mechanical factors on development, for example, embryonic development. And two, we're interested in controlling stem cell differentiation in vitro or in bioreactor. And thirdly, we're interested in how stem cells respond to the cardiovascular mechanical factors in vivo, for example, after transplantation.
So to investigate all these things, we have a custom-designed bioreactor in which we can seed cells onto elastic membranes and then stretch these membranes uniaxially for a defined set of time, defined strain, and a defined strain rate. And now we're going to step you through all the processes that you need to do to set up this protocol. Using some lab soap, any detergent will do. Get soap on both sides of the membrane and just give it a good scrub so that if there's any pieces of debris on the membrane, you're trying to clean that off, anything that would interfere with the cells. And I just do this by hand.